Hello everyone, today we'll discuss about the pharmacodynamics. So here what do you mean by pharmacodynamics? So pharmacodynamics is the study about the what drug do to our body. Okay, so what actually the drug do? It will show the effect. If you take paracetamol, what it will do? It will reduces the fever. Okay, so it will show the effect. How it will show the effect? By acting on its targets, right? So this is what we studied in pharmacodynamics. Okay, so which is the study about the effects of the drugs and mechanism of action of the drug. Mechanism of action is nothing but what? So targets of the drugs. Okay, so this is what the pharmacodynamics. So here whatever these targets for the drugs are of two types. Okay, so here, so these are the protein based targets and non-protein based targets. Okay, so targets for the drugs are the two types: protein based targets and non-protein based targets. Okay, so here protein based targets are nothing but they act by targeting various functional proteins in our body. Okay, so here nothing but biomolecules, they act by binding with various biomolecules. For example, here the enzymes, ion channels, receptors and transporters. These are the four major important biomolecules. So they acts by interacting with, by targeting these biomolecules. At the same time, only few of the drugs, they also act through their physical and chemical property. Okay, so they also act through the physical and chemical properties. So here if you take for example, if you take antacids, how they will act? So they are bases, right? After, for example, sodium bicarbonate, so after entering into the stomach, it will react with the HCl and neutralizes the HCl, thereby they elevate the pH of the stomach. Okay, so through chemical nature it will act. If you take osmotic diuretics, what are they? They are the insoluble solutes. Okay, so they are the solutes. Mannitol is a solute, so it is insoluble. Okay, and it is non-absorbable, so it will stays within this tubular fluid itself. Okay, and increases the osmotic pressure of this tubular fluid, increases the osmotic pressure. Okay, how the water moves? So water moves from low osmotic pressure to the high osmotic pressure. If the osmotic pressure of the renal tubule is increased, reabsorption does not take in place. Okay, in this way it will increases the urine output. Okay, so these are the examples. And some, if you take radioactive isotopes, so what they do? So they emit the I-131. In hyperthyroidism we will take this. Okay, so what it will do? It will emit the radiation. Okay, so up to certain distance. Okay, up to certain mm. Okay, if you take, which is concentrated in the thyroid gland, and it will emit the radiation. Okay, so and causes the necrosis of the thyroid cells. Okay, so if you take activated charcoal, so it will absorb the drug and helps in elimination of the poisoning. Okay, it will help in absorbing the poisonous substances. Okay, so these are the some example. Okay, but only few of the drugs acts by these mechanisms. Okay, chelation also. Okay, chelating agents. So these are examples. Here many drugs, they act by binding with these biomolecules. Okay, so these are nothing but proteins. Okay, either present in the cell or present on the cell membrane. Okay, so which includes enzymes, ion channels, receptors and transporters. So if you take enzymes, so we know many catalytic reactions in the body are taken place with the help of enzymes. For example, so they are responsible for the neurotransmitter synthesis, and they are responsible for the hormone synthesis, and they are responsible for various metabolic reactions like carbohydrate metabolism, and they are responsible for the cholesterol biosynthesis. Okay, so for example, if you take, so in the cholesterol biosynthesis, HMG CoA reductases help. Okay, if you want to reduce cholesterol levels in the body, you have to inhibit this enzyme. Okay, so here, so these are the importance of enzymes. That's why these becomes the drug targets, because many functions are mediated through these enzymes. If you want to increase their functions or if you want to block their functions, we have to use enzymes as a targets. Okay, so these drugs may act by using enzymes. The drugs either may stimulate the enzymes, either may inhibit the enzyme. They either stimulate the enzymes as well as inhibit the enzymes. Okay, so here this enzyme stimulation is of two types. Okay, this is enzyme stimulation and induction. Here the stimulation is nothing but, so which is rare, especially the enzyme stimulation by the drugs is very rare. But here if you give any supplements, these supplements will act as a cofactors for the other enzymes. If you give pyridoxine, for example, so pyridoxine is a vitamin B6. It acts as a cofactor for many enzymes, for example decarboxylase. Okay, dopa decarboxylase, etc. Okay, so thereby what happened, so more will be the if you give pyridoxine as a supplement, more will be the decarboxylase enzyme. It does not directly stimulating the enzyme. Okay, so here here enzyme stimulation is different and enzyme induction is different. So here the enzyme stimulation is nothing but which will increases the activity of already existed enzymes in the body. Okay, if 10 enzymes are present whose activity is increased in the present in case of enzyme stimulation. Okay, so here the enzyme induction is nothing but, so in which more enzyme proteins are synthesized, Okay, so if here the enzyme stimulation is not directly taken place with the drugs, so if you give any supplements whose effects will be increased. If you give any other drugs, okay, so that can stimulate the receptors, so receptors will indirectly stimulate the various enzymes. Okay, so in this way the enzyme stimulation will be taken place. So but here in enzyme stimulation, if Km is decreased, means Km is what here, Km is nothing but here, so here Vmax, we can in order to know enzymatic activity we can take two terms, Vmax and then Km. Okay, so Vmax is the velocity of maximum enzymatic reaction. Okay, so beyond it does not produce any effect. Okay, because all the enzymes are saturated. Okay, all the enzymes are occupied. Okay, so substrate concentration required to produce half of the Vmax is not considered as Km. Okay, so here it is the substrate concentration, here it is the reaction velocity. So to produce the maximum reaction velocity is Vmax. Okay, so to reach the Vmax, half of the Vmax, substrate concentration required is Km. Okay, so if the Km is decreased, means more will be the affinity of the affinity for substrates. Okay, more rapidly the enzyme reaction will take in place. Okay, so here in case of enzyme stimulation, this Km is decreased because the substrate affinity will be increased. We are increasing the enzymatic activity, right? So but here in enzyme induction, the Km is not affected, but Vmax is increased. Because here we are increasing the number of enzymes. Okay, so that's why here normally if 10 enzymes are present in the body, okay, this is the Vmax generally. But if we are increasing the number of enzymes, okay, so here the maximum enzyme and substrate concentration we are increasing. Instead of 10 we are increasing the number to 15, right? So that's why here the Vmax is increased. Okay, so this is regarding the enzyme stimulation. Okay, so remember these points, how the Km and Vmax is affected in enzyme stimulation. Okay, so this is unusual, enzyme stimulation is unusual, so mostly the reactions are taken place with the enzyme inhibition. So these enzyme inhibition is of also two types. Okay, competitive inhibition and non-competitive inhibition. Okay, so enzyme stimulation is of two types. here in competitive inhibition, in case of competitive inhibition, so these here the substrate as well as the drug, substrate as well as drug, both have structural similarity. Okay, for example, if this is the enzyme, okay, so this is the substrate and this is the drug. Okay, so they both have structural similarity for this catalytic site. Okay, so here both are compete for the catalytic site here. Okay, so here whoever have higher affinity, they will bind. Okay, so here drug will bind first. If we are giving it higher concentration, that's why the drug will bind. Okay, so that's why here the effect is inhibited, and the abnormal product will be formed. Okay, so this will be taken place. Here what happened here, so if here this is irreversible, here this competitive inhibition is of two types again, equilibrium type and non-equilibrium type. So in case of equilibrium type, so this it will bind it irreversibly with the drug, it will bind irreversibly with the catalytic site. Okay, however, if we increase the substrate concentration, okay, so if time increases, drug concentration will be decreased and substrate concentration will be increased, right? So whenever we increase the substrate concentration, so the drug will be replaced and substrate will bind to the enzyme. Ultimately the effect is produced. Okay, here in this case anyhow the drug is replaced with the substrate. Okay, so we anyhow Km is increased. Okay, affinity for the catalytic site here we are increasing, more concentration of the substrate is required to remove this drug from this catalytic site. So that's why here in this case Km is increased, because the affinity for the catalytic site of the substrate is decreasing, right? So that's why Km is increased, but Vmax is unchanged. Why? So if you increase the substrate concentration, ultimately same Vmax is reached, at any time the same Vmax is reached. So that's why the Vmax is unchanged but Km is increased. Km is increased means more substrate concentration is required to produce the half of the Vmax. Okay, so here another type is the non-equilibrium type. So what happened here, so here in this non-equilibrium type, so more here the drug as well as substrate both will compete for the catalytic site, but here so this drug will bind covalently, it will form a strong bonds with the catalytic site. It won't be replaced if you increase the substrate concentration, right? So it won't be replaced, it will bind covalently, okay, until the enzyme dies. Okay, that's why these are considered as suicidal inhibition. This inhibition we also called as suicidal inhibition or irreversible inhibition. Okay, then examples for this competitive inhibition, here the Km is increased and Vmax is unchanged. Okay, in case of competitive and equilibrium type. So examples, for example, if you take angiotensin converting enzyme, so this is the enzyme, angiotensin converting enzyme. Okay, so here what is the substrate? Angiotensin 1 is the substrate. What is the drug? Captopril is the drug. So both have what? Both have the structural similarity. Okay, so these are the BP lowering drugs, so they have the structural similarity. This is the inhibitor and this is the substrate. Okay, so both have competition. Here instead of angiotensin 1, drug will bind, so that's why the effect is not produced. Angiotensin 1 is not converted into angiotensin 2. Okay, so that's why the BP will lower. Okay, so here these are some of the examples. So they act by competitively inhibit the enzymes. Okay, so physiostigmine, it competitively inhibits the cholinesterase enzyme. Captopril, it will inhibit the angiotensin converting enzyme. Xanthopurinol, it will inhibit the allopurinol, it will inhibit the xanthine oxidase enzyme. Okay, carbidopa, which inhibits the dopa decarboxylase. Okay, finasteride, 5-alpha reductase. Okay, sulfadiazine, folate synthetase. So these are the examples for competitive inhibitors. So here another inhibition is also there, that is non-competitive inhibition. So what will happen in non-competitive inhibition? So here in case of drug, there is no competition between the substrate and drug because they do not have any structural similarity. Here, instead of binding to the catalytic site, so here the drug will bind to the another allosteric sites. Here it will bind. Okay, so that's why there is no competition for the catalytic site. Okay, so here the substrate affinity is normal, so that's why Km is not affected here. Okay, but here the Vmax is decreased. Why? Because so this binding of the drug to other sites will inhibit this enzymatic activity. Okay, for example 5 enzymes were inhibited, so thereby the Vmax is ultimately decreased. Normal velocity Vmax is 10 enzymes for 10 substrates per hour ankunte, here it will decreases the Vmax to only 5 enzymes per substrate. Okay, so that's why here the Vmax is decreased. Okay, anyhow if you increase the substrate concentration, it will not reverse the effect. Okay, here substrate concentration cannot overcome this type of inhibition. Examples for these drugs are omeprazole, which acts by inhibiting the proton pump; digoxin, which acts by inhibiting the sodium potassium ATPase pump; aspirin, which acts by irreversibly non-competitively inhibit the COX enzyme; then sildenafil, phosphodiesterase 5, and lovastatin, HMG-CoA reductase, acetazolamide, carbonic anhydrase enzyme. Okay, so this is regarding enzymes, how the drugs will act, either by stimulating the enzyme or by inhibiting the enzyme. Enzyme stimulation is of two types: enzyme induction and enzyme stimulation. Okay, here inhibition is of two types: competitive and non-competitive. Okay, so then other targets of the drugs are the ion channels and transporters. Okay, so these are also the proteins. Okay, membrane proteins. Where they present on the cell membrane. Okay, here coming to the ion channels, what they will do here, this is the cell membrane. Okay, they present on the cell membrane, and so they will allow specific ions into the cell and out of the cell. They will cause either influx of the ions and efflux of the ions, and may responsible for, so these ion distribution across the cell membrane is responsible for depolarization of the cell, nothing but activation of the cell. Okay, so which is responsible for muscle contraction, and these ion channels are associated with neurotransmitter release and responsible for action potentials. Okay, so these are the functions. Action potential may activates the muscle, and which activates the cardiac muscle, and which activates the which is responsible for the nerves impulse transmission, and which is responsible for neurotransmitter release. Okay, so that's why these are also becomes the targets of the drugs. Okay, but they will allow ions only across the concentration gradient. Okay, so if the ion channel is opened, only the drug will move, only the ions will move from higher concentration to the lower concentration. Okay, so if sodium channels, calcium channels are opened, they will move from extracellular environment to the intracellular environment. If potassium channels are opened, potassium is more within the cell. That's why efflux of the potassium will take in place. Okay, so here depends upon the signal they opened or closed. Okay, so these ion channels are again classified into 4 types. Okay, here depends upon, for example, some ion channels will open in response to the ligand. Okay, in response to the external ligand, for example acetylcholine binding, GABA binding, glycine binding. Okay, serotonin binding causes the opening. Okay, external ligand binding will causes the opening. Okay, so these channels we call as ligand gated ion channels. Okay, some ion channels, they open in response to the changes in membrane voltage. Okay, so for example, at particular voltage, for example, threshold voltage causes the opening of sodium channels. Okay, plus voltage is responsible for the opening of calcium channels. Okay, so these changes in membrane potential will causes the opening and closing of these channels. Okay, so that's why, so these channels we call as the voltage gated ion channels. Okay, some channels will open in response to the stretch. Okay, so these are called as stretch operated channels, and some are open in response to the changes in temperature. Okay, so those are called as temperature operated ion channels. Okay, so here in each ion channel, these ion channels are specific for specific ions. For example, so sodium channels, they will allow only sodium ions. Voltage gated calcium channels, they will allow only calcium ions. Voltage gated potassium channels, they will allow only potassium channels. Okay, voltage gated chloride channels, they will allow only chloride ions. Okay, here, so these are some of the drugs. They act by targeting these ion channels. For example, here quinidine. Okay, it is a cardiac depressant, antiarrhythmic drug. It acts by blocking the myocardial sodium channels, so this sodium influx is responsible for depolarization of the muscles and myocardial cells, and causes the excitation, increases the contraction and rate. Okay, so that's why they acts by blocking these sodium channels. Here nifedipine, it acts by blocking the L type of calcium channels. Okay, so these are either this L type of calcium channels present both on the cardiac muscles as well as on the blood vessels. Okay, so this calcium entry into this cell will responsible for the vasoconstriction as well as increased heart rate and force of contraction. Okay, so that's why this nifedipine we can use in these calcium channel blockers, we use in arrhythmia as well as in angina, whenever in hypertension as well. Okay, so this nicorandil, which acts by blocking the ATP sensitive potassium channels. Okay, so which is also used as a vasodilator. And then, so this phenytoin, so phenytoin will act by inactivating the prolonged state of the sodium channel, inactivated state of sodium channels. They will, here these ion channels, they exhibit in three phases. Okay, so here they will exhibit in inactivated state and activated state and here resting state they will exhibit, and active state they exhibit, and inactivated state exhibit. Okay, so this phenytoin will act by prolonging this inactivated state of sodium channels. Okay, so these are used in the epilepsy. Here ethosuximide, which acts by this T type of calcium channels, is present on the thalamus. This is responsible for the absence seizures. Okay, so they works by blocking this T type of calcium channels. That's why they are helpful only in the absence seizures. Okay, so this is all regarding ion channels. Next targets are the transporters. Okay, so what they do here, so these are the transporters. These are also the membrane proteins. So they are responsible for the transport of certain ions, and they are responsible for the transport of neurotransmitters, and they are responsible for the transport of various substrates, okay, across the cell membrane. Okay, so here, that's why these are also becomes the drug targets. Okay, so the here, unlike ion channels can move ions only across the concentration gradient, but these transporters, they will move the substances against the concentration gradients and according the concentration gradient. Okay, so they move from lower concentration to the higher concentration, and also from high concentration to the low concentration. And so what here, For example, it will responsible for maintaining, it will responsible for the tubular reabsorption, okay, in renal tubule, and which is responsible for the tubular secretion, organic anionic transporters and organic cationic transporter, and which is responsible for the nutrients reabsorption in the stomach and intestine, okay, amino acid transporters, so various nutrients transporters, and which is responsible for the efflux of various poisons, okay, so and which is responsible for the neurotransmitter concentration, okay, so which is responsible for the storage of neurotransmitters, okay, they will allow the neurotransmitters into the vesicle, and upon release and for the reuptake these neurotransmitters are responsible. So that's why these are becomes the targets, okay. So here, so some of the drugs, for example here serotonin, okay, after release this serotonin, okay, whenever required it will be released. This will act on the receptors, okay. After acting on the receptor, so some of the serotonin is degraded by the enzymes, okay, and some of the serotonin is taken back again, okay, after eliciting the response, okay. So this reuptake is mediated by serotonin transporters, okay. Same way norepinephrine also, after the release it will be reuptake with the help of norepinephrine transporters, okay. So here if we want to increase serotonin levels, here we have to block these transporters, right? If we want to increase norepinephrine levels, we have to block these transporters, okay. So for example here selective serotonin receptor inhibitors, okay, fluoxetine, it acts by blocking the serotonin receptors. Here cocaine, which acts by blocking the norepinephrine transporters. So for example here these are responsible for tubular reabsorption, right? So that's why if we want diuresis, we want to block these reabsorption pathways, reabsorption transporters, so thereby more will be the urine output. So here, so in case of DCT, the transporters are sodium and chloride co-transporter, sodium and chloride co-transporters. So here thiazide diuretics acts by blocking these transporters. In loop of Henle, the transporter is sodium, potassium and 2-chloride co-transporter, okay. So here high sealing diuretics, they acts by loop diuretics acts by blocking these transporter, okay. So then probenecid, which acts by blocking the organic anionic transporters. So these are the transporters responsible for the secretion, active secretion of various acids, organic acids, okay. So here in order to, here these drugs probenecid will act by inhibiting this transporter, thereby the uric acid elimination will be promoted, okay. So which is responsible for both reabsorption, this organic and also for the secretion, okay. So reabsorption is more prominent, that's why if we block this, reabsorption of uric acid is blocked, so that's why more uric acid will be eliminated, okay. So these are the 3 important targets, and another important target we have is receptors, okay. So that we will discuss in next class.